Especially.
All right.
Would you like reverse extra credit?
So
Get taken away.
so that if you, I like this thought. I think that's how
Like
pretty much
failing
everything
grade,
works.
Like permanent. Right.
All right. Chapter 16, regulation of gene expression. So this chapter is packaged in a really nice place immediately following talking about cellular communication. Because at the end of cellular communication, we talked about that the response, when the cell acts in response to whatever that signal molecule is, almost every response is the transcription and translation of some protein. And so in here, we're going to talk about regulation of gene expression, focusing primarily on what does that mean as far as the protein products. So it's basically part 2 of chapter 9. It's like chapter 9, part 2, but it's chapter 16 in this textbook, but it's not always that way. All right? And so this, you'll talk a great deal about this in genetics, which is required for some of you, not all of you. And so it's a nice foundation that you can continue to build on as you go through the program. So the first question we need to deal with is this one. How do operons provide a mechanism for altering gene expression? So we're basically going to talk about prokaryotic gene regulation, and then we'll talk about eukaryotic gene regulation. Eukaryotic gene regulation is far more complex, far more interesting, and far more relevant because we are eukaryotes and not prokaryotes. Although last time we talked about that we are actually both prokaryotes and eukaryotes, because you probably aren't human without your gut bacteria that are prokaryotes. All right, so how do operons provide a mechanism for altering gene expression? Yeah.
Just real quick, something I didn't ask last time we talked about this.
Yeah.
It was on my mind. What about newborns? They would qualify as
Newborns still have a gut flora.
at even the moment they are born?
It's not fully matured, but their immune system isn't fully matured either, but it's in place, right?
Okay.
And their brain isn't fully matured either, but it's in place, right? So there's a maturation process, but the foundation is already in place.
So there's bacteria inside their gut
Yes.
when they are born?
Yes.
Yep.
Okay.
Yep.
From their mother.
Yep.
Okay.
They get it. Yeah, they get some of it from their mother, and they get some of it from drinking amniotic fluid, which is still from their mother.
Yeah.
So,
Okay.
yep. Oh, yeah. All right. So here, focusing in on the lac operon to address this question of how do operons provide a mechanism of gene regulation. And I want you to just see a couple of key elements, and then we'll talk about the rest of it when we talk about the lac operon specifically. But the only way to regulate the expression in a prokaryotic cell is by packaging genes together with the same promoter. And so here we have 3 genes, LAC-Z, LAC-Y, and LAC-A, that are packaged together with a single promoter. And so if this promoter recruits RNA polymerase, the entire block is going to get transcribed. Meaning the entire block is going to get translated, meaning all 3 proteins are going to get produced. Okay, so it's all or nothing. You either produce all of the proteins of the operon or you produce none of the proteins of the operon. And then there's a way to control whether or not that transcription happens in the first place. Yeah.
I missed what you said in the beginning. Something about the only way.
In the beginning. Oh, yeah, the only way to regulate expression in prokaryotic cells is to package genes together under a single promoter. What do prokaryotic cells lack that eukaryotic cells have? The nucleus, right? And that's what the name means. Prokaryos means first nucleus or before nucleus. And so transcription and translation happen together. When we learned it in class, we learned about transcription, and then we learned about translation, right? In prokaryotic cells, both happen simultaneously. As soon as there's enough of an RNA for ribosomes to access, translation starts happening. So there's really no post-transcriptional regulation in prokaryotic cells. You have to control whether or not it's transcribed, and then you package these things as a unit. All right. So the way this is packaged, prokaryotes can really quickly respond to changes in environmental conditions, much more quickly than eukaryotic cells can, but the range of responses is much narrower. Okay, so prokaryote doesn't have as many different responses, but it can execute those much more quickly. And so, again, you tend to package these things together, and in an operon, you have several genes packaged together, and what you find is that each of those genes are responsible for one part of some metabolic process. Okay? Metabolic processes oftentimes require several steps. All of the proteins necessary to carry out all of the steps are all packaged together with the same promoter. So if one gets transcribed, they all get transcribed. If they all get transcribed, they all get translated, meaning you produce all of those proteins. Not you, but the bacteria in your gut. And then there are sequences upstream that control whether or not these turn on, and we'll talk about these specifically with the lac operon and then the trip operon as well. And then so there are additional proteins that bind to the regulatory sequences. We'll talk about those, and then we'll talk about the promoter and how all of this works together. But again, the take-home thing of operons is that you have several genes transcribed as a single unit, single RNA, that then gets translated into separate proteins. All right? We okay? All right. Next question we need to deal with. What are the components of the lac operon? So not just talking about operons in general, but talking about specific operons. So the lac operon is known as a enhanceable operon or an activatable operon. And so here's the lac operon. The three genes that are part of the lac operon are all associated with metabolizing lactose. What is lactose? It's a sugar, and it's a sugar found in where? Milk, the milk of mammals. And not just in cow's milk, but found in the milk of most mammals, if not all mammals, just in different rates. But lactose is a sugar. And bacteria very rarely are exposed to that sugar unless it's bacteria that live in the gut of milk-consuming mammals. And then they're exposed to the sugar often. And so these three proteins are proteins responsible for the metabolism of lactose. And so it would make sense for them to be transcribed as a unit because they're needed if they're going to metabolize lactose. They are not needed if you are not going to metabolize lactose. And so notice we have a separate gene called the lac-I, and it codes for a repressor. And so here it's written into mRNA that's translated into a repressor, and the repressor sits right here and prevents RNA polymerase from binding and transcribing this unit. So the default position of the lac operon is off. Okay, the default position is for it to be off and for these genes to not be transcribed. And that's why we refer to this as an inducible or an expressible operon. So its default position is off, but it can be induced if certain conditions are met. All right, so the default position is to have this repressor sitting here, preventing RNA polymerase from binding. So what's going to be required for the transcription, for this to shift to on, for this to be induced?
The repressor to be repressed.
You need to keep the repressor from binding, and so you need to do something to compete with that repressor, right? The repressor is a protein, and we've seen proteins before in enzymes and in receptors that they're designed to bind something. So if you can compete for that binding site, then you can repress the repressor. And so here is that repressing the repressor. So there's one, lactose comes in several different isomers, several different shapes. One of those shapes is called allolactose. And so if you have lactose present, bless you, we will also have allolactose present, and allolactose binds to the repressor just as well as the repressor binds to the operator. And so that form of lactose binds to the repressor, keeps it from binding to the operator, and now RNA polymerase can bind and can transcribe the genes that are necessary for now metabolizing the sugar that's turned it to on. So the sugar turns it from off to on, and by turning it on, the bacteria make the proteins necessary to now break down that sugar. And when they do, they'll also break down the thing that's binding to the repressor. And so eventually it'll flip back to off when there's no lactose present anymore. Is that fantastic? So, it's so cool. It's so cool. And nobody cares. Actually, that's not true. I'm not talking about in this class, because obviously if you didn't care, you wouldn't be here. But I'm talking about hardly anybody does research on gene expression in prokaryotic cells. It's so sad. Everybody wants to do it in eukaryotic cells. All right, so to sum this up, the lac operon has 3 genes, lac Z, lac Y, and lac A. And again, those 3 genes are associated with metabolizing lactose. Default position is in the off position. And so it is what you would call negatively regulated by the repressor. And so its default position is off, but when lactose is present, you have some allolactose that interferes with the repressor and switches it to on. And so there's a separate gene that codes for the repressor. And so this is an inducible operon, and again, induced by the sugar that needs to be metabolized. All right, so I have a question. We're going to do ourselves a short lecture break. Hopefully you're still roughly in the same groups that you were in for your quiz. And here's a question. I'll give you about 60 seconds or so to talk about this. What would the impact be of a mutation in the lac operon? So if you had a mutation in the operon, what would the impact of that be? And how would that impact vary by the location in the operon? All right, so think about what's part of the operon and how a mutation would impact the bacterium. All right, take about 60 seconds starting now.
We'll be able to break down lactose, and if it's an addition promoter, then it would either have too much or too little of it, and if it's addition of different parts of it, it will deal with whether it can break it down, basically.
Yeah,
Lactose is
so it would be lactose. Well, are there like
The answer to your question is yes, Grace. There are. Yes. Yeah, very few human beings can actually metabolize lactose. Most of the time, yes.
So it's not even my fault if I'm lactose intolerant that I can't digest lactose.
Correct.
They're not pulling their weight.
Are you lactose intolerant?
No, I'm allergic to milk, but I'm not like, I think it's lactose intolerant. It's like,
You're allergic to the milk protein? Probably casein.
still though,
Then it's just gonna happen when the proteins are
what bacteria to pull their weight?
so different parts of the process of digesting lactose.
How many people not eating cheese?
All right. So let me ask you this.
And if it's promoter, it's gonna have like too little.
If the mutation is in the last gene of the lac operon, what are the potential impacts of that mutation? Potential impacts. So it's in the third and final gene. So it goes lac Z, then lac Y, and then lac A. So the mutation is in lac A. What are the potential impacts?
It can't digest stuff.
Well, that whatever that protein does, it can't do that, right? So overall, you're probably going to inhibit the metabolism of lactose, but you don't know how far in that process you're going to get, right? You'd have to know what each of those genes do, but that's only one potential, because remember there are silent mutations. So that mutation in the lac A gene could lead to the same amino acid being put in the correct spot. It could be silent, or it could even lead to a different amino acid, but the amino acid is chemically the same, so the protein functions the same. Or it could alter the impact of whatever that protein does, at which point you probably wouldn't be able to metabolize lactose, but it depends on what part of that. Because what's required to metabolize something? Thinking that the only way a bacterium can metabolize something is if it ends up inside of the cell, right? So what does it need to do to metabolize it? It needs to get it in the cell, and then it needs to break it down chemically, and then it needs to harness whatever that energy is that's released by breaking it down, right? And so whatever that gene does is going to be inhibited by that process. If the protein or if the mutation is further upstream, like if the mutation is in the, say, lac Z portion of the lac operon, then is it going to be that protein or all three? Depends on the type of mutation, right? If it's just a point mutation, it's just going to be that one, right? But if it's a frame shift mutation, then it's going to impact every gene in the lac operon. Yeah.
But if it's the last one, then it will only impact the ones after, not before it, no?
Well, correct. But it would impact the gene where the mutation is. But nothing before it. Nothing
So
upstream.
if it's the very last one and it's frame shift, it's a less big of a
Yeah, but it still inhibit the protein activity of the last protein. Correct.
It's the very last thing, the very last thing. Even if it's frame shift.
Sure. Yeah. And then if the mutation is in the promoter, the entire thing is messed up. And what's even more interesting is what if the mutation is in the operator? What binds at the operator? The repressor, right? So if the operator is mutated, the repressor can't bind, and now instead of the default position being off, the default position is on. It's very wasteful because you're set to metabolize something that may not be present, right? Okay. That's a good question for the final exam right there. That's a really, really good one. Just, putting that out there. So important questions. The next one. When is the lac operon active? Somebody answer this for me. When is it active? When the repressor is repressed. Okay. Any other ideas? So, and that's going to happen when lactose is present.
When you don't lack lactose.
When you don't lack lactose. But do you imagine that that's it? So if lactose is present, the bacteria are going to flip this to on all the time. What if there's something easier to metabolize? Right? So lactose is a complex sugar, which is why when bacteria in your gut can't break it down, it causes you so many wonderful feelings, right? It's a really complex sugar. It's big and it's bulky and it just clogs things up, right? It's not good. Or it does the exact opposite and induces a lot of water to flow out of epithelial cells lining your gut into your tract. Either way, it's not good, right? Not a good situation. It's a very complex sugar. So if there are easier sugars to metabolize, even if lactose is present, it would still make sense for them not to turn this on. Okay, so in addition to inhibiting the repressor, you also have to enable RNA polymerase to bind. And what you find is that RNA polymerase doesn't bind unless glucose is also absent. So you need lactose to be present so that you inhibit your repressor. And you also need glucose to be absent so that you activate this cap protein that gets RNA polymerase to bind at the promoter. And so this will only bind when cyclic AMP is active, and that's only active when glucose is absent.
So the cap's default position is on unless there's glucose?
The cap's default position is inactive. So this is the default position of this cap protein is inactive. The default position of this is to have the repressor binding here. So the default position is glucose is present, lactose is not, right? Lactose is not present, your repressor's bound here at the operator. Glucose is present, this protein is inactive. And even if glucose isn't present, this would now be active and would help RNA polymerase bind there, but your repressor's sitting there. So you need glucose to be absent and lactose to be present in order to fully switch this on.
What I mean is the cap all by itself without anything is active unless you bind glucose to it.
No, the cap is completely inactive unless glucose is absent.
How does it know?
Because when glucose is absent, the cell produces a molecule called cyclic AMP, which is a nucleotide monomer. It's AMP is adenosine monophosphate, just like ATP is adenosine triphosphate, but it's this particular form of that, and it's an indication that the energy components or the energy source within the cell is extremely low, which usually means that glucose, which is the most readily available of all the sugar molecules, is absent, and the cell is starved. So basically, in order for this to flip fully on, the cell needs to be starved and lactose has to be present. Yeah?
Does that mean, like, even if you're not lactose intolerant, if you eat sugar with your cheese, you're going to have a harder time digesting the cheese?
Yes. Yep, you'd have a harder time digesting the cheese.
So cheesecake is not good.
If you Oh, cheesecake is good. It's very good.
Counterproductive, though.
Your gut bacteria tend to vary based on your diet. So if you've got a very heavy lactose diet, you'll tend to develop more bacteria that will metabolize
So
lactose.
favor those bacteria basically.
Yep. Yep. All right, so sum this up. When is the lac operon active? It's active when lactose is present and glucose is not. So when glucose is not present, the cell is starved and it activates a protein that will enable RNA polymerase to bind. So we have a couple of questions we need to deal with. We'll do, again, one short lecture break. Why wouldn't the lac operon be active when glucose is present? So even if lactose is present, why isn't the lac operon active? Why does glucose have to be absent and lactose need to be present? And then another one, which is a more interesting question, is how can a bacterium get this operon if it didn't have it from the beginning? What are some ways in which this bacterium could get this operon? Okay, go ahead and take about 60 to 90 seconds, talk this over with those around you, and we will talk about this together. 60 to 90 seconds, starting now.
Because glucose is easier to digest, and so it's a more, it's also it's just more energetically
Efficient. It's fine.
Do you guys have ideas?
So I think some genes are like representative of elements.
I haven't been able to write and read in this class in a long time.
So it's a mutation in glucose, probably a mutation in like plasmidine, and then they just like share it. Well, this is
Or it could be like conjugate, like when
the
you
lactose.
bacteria, let's swap genes for fun.
The lactose.
Yeah, that too. Probably how it already could do glucose.
Right.
Or like it would bring it apart, like, oh, there's glucose and then there's lactose. It's like, what do I do with this lactose?
But it probably was a mutation in the glucose one, and then combined the glucose with another glucose, and then like mutated a little bit, so it can now
They do like,
and then it transfers to like shared multiple bacteria.
I don't know if it's gonna be this kind of mutation.
Also, there's enough glucose already. You're not going to want to separate
All right.
the lactose.
So,
Lactose.
in case you are unaware, lactose is a disaccharide, a sugar made up of two monosaccharides, and it's made up of glucose and galactose. So in the process of metabolizing lactose, you still have to metabolize glucose. Right? So part of the metabolism is to cut this, and then you have glucose, and galactose metabolism is very similar to glucose metabolism. You can shuttle galactose into the glycolysis fairly easily. Okay, it comes in pretty easily. It's not really complicated to convert this into glucose and shuttle it in. Actually, I think you convert it into fructose, but that's like, step 3 of glycolysis. And so, in the process of metabolizing lactose, you still have to metabolize 2 molecules of glucose. So the glucose metabolism never stops, even when the lactose metabolism starts. And so, the cell's already positioned to metabolize glucose. There's really no need to metabolize lactose, even if it's present. Plus, it's complicated to get it into the cell. It's big and bulky, and actually requires effort to get it into the cell. Okay, it doesn't diffuse as easily across the cell membrane as glucose does, and so it's complicated to get it in. Now, how can the bacterium get it if it didn't already have it? Yeah. I heard some people talking about viruses, right? Where was that? But you had a thought, Alyssa?
Oh, I just think maybe because it already has the glucose, the way has a way to metabolize glucose already, and that's kind of similar.
Okay. So building on something that's already present.
Yeah, so it probably had 2 of them together to it copied the thing of how to do glucose twice accidentally, and then kind of mutated and that works, and then it should both bacteria friends.
Yeah. Now you would think that, but metabolizing lactose is significantly more complicated than metabolizing glucose twice. Right? It's like so I mean, let's say I don't know, let's say you were going to play a 4-year-old in a game of cornhole, right? Do you know what I'm talking about with cornhole?
Yeah.
Okay, it's beanbag toss. You got a hole in a board and some beanbags. Like, I will crush my 4-year-old in cornhole, no problem. You add a second 4-year-old, I'll crush them both. It's no big deal. But it's not like that. It's like if you could get 2 4-year-olds together, it creates something unique and challenging that you have to deal with. Which I guess they could do that, but 4-year-olds are very easy to manipulate. They don't think very hard. But it's a little bit more than just metabolizing glucose twice. It's fairly complex, which is why we can't metabolize lactose, even though we metabolize glucose in every cell in our bodies. But very few human beings can actually metabolize lactose. It's fairly complex. What was your idea of viruses? Share us your wisdom. Who is mentioning viruses?
I thought they inserted some kind of gene way to
Uh-huh.
make the proteins that would allow them to do this.
Yeah, it's interesting. Viruses do that all the time with bacteria. Viruses change bacterial genomes constantly by injecting genetic material into bacteria. It's called transduction, and bacteria do it. I mean, bacteria are transduced by viruses constantly, and viruses manipulate bacteria. And what you find is the bacteria in our gut are heavily manipulated and controlled by viruses. And so just in terms of number, you have far more bacterial viruses in your body than you do animal viruses. And most of those bacterial viruses are controlling your gut bacteria and keeping them from growing out of control, and even manipulating them and influencing how they metabolize. So that's one way, but bacteria can also transfer genetic information one way. It's called conjugation, and they will willingly give material to other bacteria. We talked about that, and maybe we didn't. I don't know. Yeah, listen. Yes. Yep, it's one example of horizontal gene transfer. And then another way, bacteria just take on genetic material like they would any food source. But then as they take on that genetic material, it tends to form a circular plasmid that they don't metabolize, and they just keep and then transcribe, whatever's on it. So you can induce bacteria to do that as well, which is nice. So if you ate an animal that had bacteria that can metabolize lactose, it's possible that those bacteria could contribute that genetic information to your gut bacteria. Right? All right. So the next question. What are the components of the trip operon, and how does it differ? So what did we call the lactose operon? What type of operon is it? What's a lac operon? But I called it an inducible operon because its default position is off. Okay? The trip operon is called a repressible operon because its default position is on. And so the default position is on. And in this operon, it's basically the operon gives the bacterium the ability to make tryptophan. Tryptophan is an amino acid used as the bacteria translate mRNA into protein. And so if tryptophan is not present in their environment, they will make it. And tryptophan is a fairly rare amino acid. And so most of the time they are not exposed to tryptophan. And so the default position of this operon is on, so that they are making tryptophan. This one has 5 genes in the operon instead of 3. And again, the default position is on. So the default position is not to have the repressor bound to the operator. And so instead, you have this regulatory gene that's making the repressor in inactive form. Whereas in the lac operon, your repressor gene, which was lac-I, was making the repressor in active form, and it had to be deactivated by something being in the environment. And this has to be activated by something being in the environment. And so if the default position is on, and when tryptophan is absent, what do you imagine activates this? Tryptophan, right? So in the absence of tryptophan, the default position is on. The bacterium is making tryptophan. If tryptophan is present in the environment, it's energetically wasteful to make it. And so tryptophan will activate the repressor, which will then allow the repressor to bind to the operator and to shut down the transcription. And so this is a repressible operon. The default position is on. and the repressor protein is activated by tryptophan. When tryptophan is present, repressor protein is active, binds to the operator, transcription shuts down. So how does it differ from the lac operon? We'll sum this up. The operon, it's repressible versus an inducible operon. Has 5 genes instead of 3. Something in the environment activates the repressor to turn the operon off, versus with lac operon, something in the environment deactivated the repressor to turn the operon on. And then we have one more question that we're going to deal with. And so I want you to take about 60 seconds, talk about this with those around you. Which operon, the lac or the trip operon, is more complex, requiring higher levels of control? And so this is another really, really good question. Just putting that out there. So I liked Alyssa's idea. I don't know if y'all heard it, but could I put some bonus questions on the final exam that cover chapter 16? I like that idea, and I really like this question. Just putting that out there. All right, take about a minute, talk this over with those around you, and then we can deal with this. Which operon is more complex?
It's the lac one because it needs 2 things.
It needs 2 things.
This one just needs 1.
Oh, because it needs the lack of.
Lay it on me, Biggs.
And also the lack of
It's the
has to make something else be made, which then makes that repressed. This is just like the thing that it makes represses itself.
They're
It's really like a little circle.
So it's just its own.
It's like, but they're all expressed together.
So tryptophan.
It's not really the size of it,
TRP is
it's expressed
dependent
as
only on
well.
tryptophan either not being there, whereas the lack operon needs lactose and with the, and on top of that with the glucose.
Well, it makes sense
it's
because
inactive.
so the tryptophan regulates itself so much. Tryptophan is produced, says, don't produce
It's kind of like
there's no tryptophan, produce tryptophan. Whereas the lactose is like, lactose is
It has to
present,
make its own thing.
produce enzymes, enzyme digests lactose, lactose isn't present.
It has to make it.
You know, there's like, it's not producing what's inhibiting it, it's producing something that digests something else.
And it actually has to be like turned on.
Yeah.
It has to have a yeah.
And then with the lactose, you have like repressor, and this one you just have it on, it's just like on.
Not necessarily.
As opposed to off.
Yeah.
All right.
I just feel like since they're all expressed together.
Any thoughts on which operon is more complex, meaning requires higher levels of cellular control? That's the key part of this question here, requiring higher levels of cellular control. Yeah, Molly.
We said lack because there's only 3 structural genes.
Mm-hmm.
So to keep it from having mutation, there has to be more control in those genes.
Potentially.
Whereas in the trip, there are 5 genes, and so there's not as much. There is more room for, or there's not as much room for mutations, I guess, because there's more genes there.
Maybe more control. Maybe it's potentially more redundancy.
Yeah.
Yeah. I think you have the same idea, but for a different reason.
Oh no, I was thinking lac is more complicated because it takes 2 different things for it to be activated or inactivated.
Yes.
And the second one has like 2 steps to it. First has to make the cyclical EMP, and then that has to block
Right.
that. And then so it's just like a bunch of different processes that could potentially go wrong versus just one.
Yeah, it's certainly investing more control, right? Because it doesn't need to just have lactose present, which inhibits the repressor, but it also needs to activate the cap protein that enables RNA polymerase to bind.
And to enable it and to activate it, then it has to.
Sure.
do that.
And so there are 2 levels of control rather than a single level of control. And 2 is greater than 1, right? And so that's a nice question because I think it emphasizes not only the difference between the 2 types of operons, but also how much the cell is investing in ensuring that process doesn't happen. And the cell's investing a lot in ensuring that it doesn't transcribe genes for metabolizing lactose. And that's interesting to think of. Why would it invest so much energy to ensure that it doesn't make 3 proteins, right? There's got to be something more than just energetic wastefulness, because it's investing more than what it invests in not making tryptophan when tryptophan is present, and that's 5 genes, but it invests more control to not make 3 proteins than it does to not make 5. So there's more than just energetic concerns. Yeah.
So maybe those enzymes have other side effects? Like maybe they digest other things that shouldn't be digested?
Yeah. No, it's exactly right. One of the proteins that it makes digests almost any disaccharide, and some of the disaccharides that it digests produce sugars that are toxic to the cells. Yeah. So, all right. Any questions, thoughts, concerns, things you feel like you want to share? We'll go ahead and call it quits here and put a little bow on our Friday. All right.